Hey everyone, Mr. Murphy here, and I'm going to be going over section 7.2, which is all about mitosis. So there's four main things to remember that need to happen in the process of splitting the cell. First, there needs to be a signal, a reproductive signal of some sort that tells the cell that it is in fact time to divide. There's then the replication of DNA. This is important because you need both of the new cells to receive a full copy of the DNA. The process of segregation is the distribution of those DNA copies. You need to, again, make sure that the appropriate sets of DNA end up on either side. Otherwise, you could have, duplicates of certain parts and be missing other parts of the DNA. So it's not just like double the chromosomes and then sort them out 50-50. It's double the chromosomes and make sure one complete set gets to one side and one complete set gets to the other. And then the final step is cytokinesis, and this is the actual karate chop splitting of the cell. The process is a little different in prokaryotes because they don't have a nucleus. So we refer to this process as something called binary fission. And what's happening here is the cell is going to grow, first off, because any cell that's dividing is going to need to be twice as large so that when it splits, it's now two full-size cells. Now, it doesn't have to be twice as large before the split occurs. Of course, cells can get a little bit smaller and then individually grow back to their original size. But in addition to growing, it needs to replicate its DNA, so just like in eukaryotes. And then this is the splitting of that DNA and cytoplasm into two cells. So for prokaryotes, a way in which it's a little bit different is that they typically only have one chromosome. These are very simple organisms that don't have nearly the complexity and efficiency of a eukaryotic cell, and so one single chromosome in a circular fashion. And thinking about that may seem strange, but of course the nature of chromosomes in our body is also very complicated, and the shape of it is by no means simple. In the case of a circular chromosome, basically, ordinarily you have just this line of coiled together nucleic acids. In the case of a prokaryote, the bottom portion of that just feeds back into the top, so it's a closed loop, which adds some structural stability and protection in the absence of a nucleus and additional membranes to keep it safe. So some terminology that comes up, you'll hear about the ori, which is the point of origin, the place where replication starts, and then the ter region, which is like the termination region, and that's where replication ends. We specify this simply because in the case of a prokaryote, you have a circular chromosome. It's hard to designate like what's the beginning, what's the end of a circle. So the ori is going to be the point at which the replication actually begins. The chromosome begins to unravel. The two strands of the double helix of DNA begin to separate and allow for replication to occur. So this is at a site that we call the replication complex, and that's just basically a term referring to all these proteins that are facilitating it. Something we'll get into in the genetics unit is talking about polymerases and helicases and these different enzymes that facilitate the individual little steps that need to happen in DNA replication. So as this occurs, the ori is the point at which at first the double helix splits apart and the two sides begin receiving their complementary pairs to create two new copies. And that all requires energy. There's ATP used for the entire thing, and there are cytoskeleton components that are facilitating the movement of this. So getting ahead of ourselves a little bit, but we know mitosis involves these spindle fibers that pull the individual chromatids to either side of the cell. Very similar functioning going on inside of a bacterial cell, as the two now replicated circles of DNA are going to need to end up on opposite sides. And here you can see an image that tracks the ori here with this red dot. You can see how as it begins to replicate, there are now two of these red dots signifying basically that origin point of both of the now replicated copies, and those red points are getting pulled to the side by unpictured cytoskeleton components, actin filaments that are using motor proteins to pull these things to either side, and then by the time the cell splits, each side has a copy of the original DNA. And you can see both of these are, absent the color, which is just used to designate the difference. It is the same size and shape and containing the same DNA as the original cell. So cytokinesis begins after that separation. Think of it as just basically the pinching together of the plasma membrane. It's a little different in plants because of their very rigid cell wall, and we'll see why that's, or how that's a little bit different. In the case of bacteria, they also have a cell wall. However, it's not nearly the rigidness of the cellulose-based cell wall that plants have. So not something we need to worry about, but of course that is happening in the background here as well. Eukaryotes have to undergo the process of mitosis in the midst of this. So there's replication of DNA, and that's only going to happen at a specific point right before the cell is going to replicate, followed by mitosis, followed then by cytokinesis. During the separation, the segregation of that DNA, one copy of every chromosome is going to end up in each of the new cells. So that way, if you have just a regular human body cell with 46 chromosomes and it's going to replicate, we need to make sure that each of the daughter cells receives 46 chromosomes, one complete set of chromosomes each. An important term, and I kind of give everyone grief about this in class a little bit, mitosis is not the splitting of the cell. So we have to understand that mitosis is the splitting of the nucleus, which is important because we see mitosis in eukaryotes, which should make sense immediately, that anything that's related to the process of mitosis is going to be found within all eukaryotic organisms, because if you have a nucleus, you're a eukaryote, and anytime you split your cell and need to make identical copies, you need to split that nucleus entirely, which is going to require the process of mitosis. So then cytokinesis is going to be very similar to the splitting of any other cell. And then, like I said, we'll talk about plants. So I'm going to go through this kind of quick. This might be something that you need to periodically pause and go back and think about some of the terminology that comes up. But just understand the process is very straightforward. Individual steps are pretty logical, governed by different cytoskeleton components and things like that. So there's all sorts of chemistry and things happening in the background. But don't get thrown off by sort of the volume of material that is being discussed. It is taken in pieces. It's a relatively simple process. All right, so during interphase, which is the vast majority of a cell's life, there are three what we call subphases, and there's two gap phases and a synthesis phase. So G1, S, and G2, and they go in that order. So G1 it's a growth period when the cell could just be hanging out, just doing its normal cell things. And this is also, as we'll see in the next section, this is also the place where some cells just sort of come to rest and will simply just do their thing, and maybe they're not actively dividing all the time. And then once the signal's been received to actually begin the process of replication, the cell moves into the synthesis phase, because the first thing that needs to happen is we have to make a new copy of the DNA. The sort of most important component of successfully splitting a cell is making sure that each of the new daughter cells get a complete and undamaged copy of the original cell's DNA. The G2 phase is another period of time that happens after the synthesis and before mitosis actually begins. So this is like final preparations. This is making sure that the S phase has occurred correctly, and just generally allowing the cell the time to get the parts in place to allow for mitosis, because we'll see the cell's doing a pretty drastic thing in the midst of mitosis, as it's going to break down its nucleus, separate these chromosomes, and then rebuild the nuclei on either side. And of course that needs a lot of cellular machinery in order to happen correctly. So here you can see kind of the more stereotypical, like when you think of a chromosome, you think of this like X-shaped thing. This is actually two copies of a particular chromosome. So this is a replicated chromosome, and you could think of it as being like this one little thing on the left and one on the right, and then they're sort of stapled together with this structure called a centromere. So when they're in this form, we call them sister chromatids. You might hear it referred to as a replicated chromosome, but this is actually two copies. So this is after S phase. You really would only see this in the midst of mitosis in this X shape. And here we can see a cell which is beginning to condense its DNA within its nucleus. So here's a good representation of how this process goes, and we always represent mitosis as being this really small period because for a typical cell, it's this very small blip in the history of what that cell is actually doing. And this isn't really realistic at all because many cells will have a G1 that is 99.99% of what their life is. But for the sake of just arranging this information, this is a helpful thing just to clue us into the fact that interphase is really long, mitosis is relatively short, and this dark blue line here is cytokinesis, that final chop of the cytoplasm in order to split the cell into two new ones. Represented here is the single copy of DNA that exists in the cell during G1, the fact that there are two copies now in G2, or by the time G2 starts, after S phase you have two replicated sets. Okay. Mitosis produces two daughter cells, and so the cell cycle produces two daughter cells. In mitosis, it produces two daughter nuclei. So at the end of telophase, as the cell is beginning to pinch apart, you have two nuclei inside there, and that's why when cytokinesis happens, we're good. We have one nucleus in one cell, one nucleus in the other. Mitosis is continuous, and what that means is we learn it as prophase, metaphase, anaphase, telophase, but it's not like prophase and then the cell stops and looks around and declares like prophase is complete. It's a gradual process. All of these steps are happening on top of each other, but breaking it into these different subunits is a helpful way for us to understand the intermediate goals that need to happen in a particular order in order to successfully split the cell. So during interphase, you have the nuclear envelope. The nucleolus is inside of that. That's probably the only visible components that are really occurring inside of a cell if we were to look at it in the microscope. We can't really see the DNA yet because the chromatin's not fully condensed into the chromosomes. So during prophase now, this is where things begin to visibly change about what's going on inside of the cell. We can see the condensed chromosomes. We can see the centrosome, which we'll talk about in a second. We can see the spindle fibers beginning to form. The condensed chromosomes are the chromatin wrapped up with all sorts of proteins into this tightly packed thing that's going to be easy to manipulate within the cell compared to, this long free strand of millions of nucleotides. Sister chromatids are going to be the two halves of that X-shaped chromosome, and that is the replicated copies of the identical genetic material for each of the chromosomes. So inside of a human cell that is dividing, you would see 46 of those X chromosome shapes, but understand that those X shapes are each two copies. And so by having 46 of those with two copies each, when those chromatids split apart, each cell is going to get 46 individual chromatids, which is 46 chromosomes. It gets confusing because we use the chromosome word in a variety of ways, but we want to be thinking about it in terms of making sure that both sides get a full, complete set of the DNA. The centromeres where they're linked together, we saw that before, and then kinetochores are the proteins that are actually present on the centromere, and that's where the spindle fibers are going to be attaching and able to basically apply an actual physical force to either side in order to pull those chromatids apart, breaking apart the centromere. In reality, that's mediated by many more proteins that are going to be facilitating the separation of those chromatids. A karyotype is a word to refer to basically our view of the full, complete genetic material of an organism. So we can analyze karyotypes in order to understand the distribution of genes within an organism for a whole variety of purposes, more and more purposes every day as we get better at understanding the full genome of organisms. Here's just a picture of the human karyotype, and you can see it has the 22 what we call the autosomal chromosomes, and then we have our one sex chromosome here, which is the X or Y. And so this would be inside of you or me, we would be able to find these chromosomes, two copies of each chromosome, one that you received from your mother, one that you received from your father, and then all of these. And if you were a male, you would have one X and one Y, and if you're a female, then you'd have two X's. The process of segregation is aided by also the centrosome, not to be confused with the centrioles or the centromere. So there's a lot of terms that are very similar to each other because they're related to this same idea. But the centrosome is actually the region, or not region, the collection of two centrioles that are going to migrate to either side of the cell and begin producing the spindle. And the spindle are microtubules that are going to reach out, grab onto the chromosomes. There's other microtubules that reach to each other in the middle and will begin pushing against each other. And then there are microtubules that grab onto the opposite sides of the cell and begin to pull all of it apart. So the stretching of the cell, the pulling of the individual chromatids apart, all of that is mediated by the spindle, which is produced by the centrioles, and the pair of centrioles is referred to as the centrosome. And there you go. There's the terms for you. So this is just what I was mentioning. Polar microtubules, those overlap in the middle. They're like the pushing apart of each other to push the centrosomes apart on either side of the cell. The kinetochore microtubules are the ones actually attaching to the kinetochore and pulling those chromatids apart. During prometaphase, so all of that's prophase. During prometaphase is when the nuclear envelope breaks down. Now, this is a phase that we sort of sneak in. Surprise! There's a bonus one. It's AP Bio, so we don't just say prophase, metaphase, anaphase, telophase. Now we say prophase, prometaphase, metaphase, anaphase, telophase. It changes nothing. It's a continuous process, like we mentioned before, but this just basically gives us like another sort of step-by-step things that need to happen in a particular order. In some textbooks, you'll see things referring to early prophase and late prophase. Instead, here, our textbook's just designating like there's prophase, and then there's prometaphase, which is kind of the late prophase before metaphase thing. So here we have chromosomes consisting of two chromatids, identical chromatids, attached with their attached to the kinetochore microtubules. So the microtubules are attaching to these chromosomes now during prometaphase. Nuclear envelope's gone. I kind of went through that quick, but the nucleus, gone. That's a pretty big moment that now this is a cell that has no nucleus. So if you're wondering, like, is this a slow process? Is this a fast process? It's got to be pretty quick from here on out because we no longer have a nucleus. We no longer have accessible DNA. It's all packaged up, so we can't be making RNA copies. We can't be doing a lot of transcription translation. The cell's kind of in pause mode for right now. It can't address its homeostatic needs because it's in the midst of reshuffling everything that's going on. And so, as you would hopefully imagine, mitosis is relatively quick no matter what cell it's happening in. The cell can't afford to just hang out in the midst of mitosis. So here's a good visualization. We have interphase, everything's just normal, you can't see any chromosomes. During prophase, the chromosomes condense, or the chromatin condenses into these chromatids, and then they have linked together. These homologous pairs have found each other. We're beginning to get the spindle, and then now during prometaphase, you see the nuclear envelope has gone away. The spindle fiber is now attaching to the kinetochore on each of the chromosome pairs. During metaphase, it's the easiest phase. They line up in the middle. Metaphase, middle. They line up those chromosomes in the middle. During anaphase, also easy. We begin to pull the chromatids apart. So anaphase, apart. This is how I teach it in my beginning bio class. It keeps it easy. Middle metaphase, they pull apart in anaphase. After they are separated, the chromatids, they then get pulled to either side to where the two daughter cells will begin to form. So here we have metaphase lined up in the this plate down the middle, this hypothetical line in the middle of the cell, and then those kinetochore microtubules begin to sort of reel them in. And there's all sorts of other proteins involved in that separation, as I mentioned before, that pulls them apart. And then we end up, during telophase, we begin to make two nuclei. So two nuclei in telophase. Middle, apart, two nuclei. Easy ways to remember these things. There's a little more detail that you won't get quizzed on, but there are, kinesin and dynein motor proteins involved in this process. And these are, we watched like the Life Inside of a Cell video where there's a little motor protein dude walking along a microtubule. That's dynein or kinesin, which are the two main types or families of these motor proteins that actually do the movement of things inside. Requires ATP through the whole thing. There's a pretty substantial and significant event happening inside of the cell. So while we don't talk about it directly step by step, just understand that ATP is being chewed up through all of this which again is why it needs to happen pretty quick, because the cell's not maintaining that supply of ATP nearly as efficiently in the midst of splitting the cell. Telophase, we already saw the picture, and really it spells it out. The spindle starts to break down. We've succeeded in pulling the chromosomes apart. The chromosomes begin to uncoil as the new nuclear envelope surrounds them on either side, which now we're reaching that success point. Cytokinesis comes in. That's the division of the cytoplasm, so the splitting off, the pinching together in animal cells. That happens because of a contractile ring, which is just microfilaments and proteins just pulling together and pinching off the two until the membranes represented by my index fingers meet up with the membranes represented by my thumbs, and then some proteins facilitate basically swapping and pulling off, and now they are separated. Here you can see a picture of that. This basically squeezes together until it splits. In plant cells, it's really cool, but it's also pretty simple or straightforward. The Golgi starts churning out all these vesicles, and those vesicles then gather up across the midplate of the cell and start to basically bubble together. Proteins involved in sort of allowing those membranes to stitch together, but it makes this new membrane right across the middle of the cell, and then at that point the cellulose begins to form the new cell wall, and you end up with two distinct plant cells. So it's kind of cool. The cell grows and stretches, and then grows this down its midpoint. It grows this new membrane and new cellulose. After cytokinesis, congrats, you have two cells. They are identical. Everything about them should be the same. They have even numbers of chromosomes in each. That is the normal somatic cell set of chromosomes. There's more to, what's actually happening. Are you just replacing cells? Are you making new copies of cells that are going to go and do things? Or are you building tissue cells? Because then orientation and things like that, where these cells are growing to form a larger structure like tissues or organs, that's gonna be sort of a differentiating factor. There you have it. And that is mitosis. Don't forget binary fission, which is what we started with. And in the next one, we will talk about how this whole process is actually controlled. Let me know if you have questions.